Hello everyone. This is the Asia Pacific Research Platform, the working group session 1. So I'm Jung-hoon Moon from KISTI in Korea. The KISTI is the Korea, the representative of the Supercomputing Center and the Research Network Center. The miking is a little bit howling. Please reduce the volume. And the first session is I want to very shortly introduce about this APRP working group. APRP working group is initiated at the APAN 45 meeting in Singapore. At the time, and so we made the birth of fees, and then we are going to be to the working group, the formally working group in APAN. So the name is the Asia Pacific Research Platform. The research platform comes from UC San Diego and the ESNet, and it comes from the US. They have, is it okay to the howling? There's a howling. Yeah, the sound is howling. That's okay? Okay. And yes, it's basically the technology based on the science change technology. It means that is the very ultra high speed, the big data superway, and the combined the distributed the computing source, using the Kubernetes. The Kubernetes is well known as middleware to gather the computer resources to make one thing as a part and to provide to the application users. So our working group, the object is the share of XIP. X meaning is the ARP, the NRP, PRP, or the X can be the anything. And the Korea, in case of Korea, KRP. Australia is ARP. New Zealand is it just assumed that NGRP. Yes, something like that. So we want to promote the HPC ecosystem in the Asia Pacific regions and engage APAN members and the ASEAN countries, and then toward the setting of an Asia Pacific research platform and become a part of the global research platform. The global research platform is the totally wanted to make this one things for globally. So it's kind of the consortium, and the XRP, the readers and the engineers as members come join to the GRP workshop and share their experience and how to promote this in the future, the local areas. Our target is academia and the industry. Executive member is chair and the co-chair, and we have the secretaries. And then the particular thing is we they get funded from this 10 office the last 1 year, the building a high bandwidth distributed HPC. I will explain about this 5 minutes later. So, and we are the last 6 years, we are the 12 times for the 6 year held working group meeting and 24 sessions in several countries and several presentations. So I will shortly introduce about this, the session 1. That we have the 8 presenters. Some of the presenters is from remote, some of in presence in here. The second session is the chair is Andrew Howard from the NCI in Australia, and the second session has the 6 presenters from the remote and on-site. So I wanna give a talk to the first program is the Asia Connect project, building a high bandwidth distributed HPC. Actually, this the presentation is the last on Monday, and is I presented the 10 Asia Connect meetings and for deeply. I just briefly introduce about all of these the overview and the review and very shortly in this time. So as I mentioned before, the APRP working group is funded from this 10 office. The name is Asia Connect Project, and the title is the Building a High Bandwidth Distributed HPC. And the project overview is just 1 year, and the budget is 150K euro. And the participant countries is Malaysia, Korea, Pakistan, and Australia. The participant countries' role is Australia and Korea is donated our experience and donated some technical issues. And Pakistan and Malaysia donated the local science application. In case of Pakistan, it's AI science. And in case of Malaysia, they apply for this bioscience. They have a lot of the bioinformatics scientists and areas. In case of Pakistan, they have a lot of the students and the members for the AI areas. And then this is the project background. We can find out is the left picture is the PRP project. It comes from a Pacific Research Platform in UC San Diego in US. They make this the white line is the big data superhighway on the California research network, and the blue spot is the science DMG based DTN node, and the research platform is combines this infrastructure and made by this Kubernetes. And the Kubernetes is the managing this distributed CPU, GPU, and the memory intensive, such like servers, and make the one resource part to give this provide what they want to use. So at that case is the very the remarkable the result to the CO2 the researchers depends on this climate change, and they are using a distributed for the locally distributed for servers and make one resource part and running their jobs, and the result is remarkable. And then left side is in case of Korea, the apply for this LSTM, the AI, the editable, the algorithms to deploy in the 24, the Korean, the government funded research institute. Those results is the very insightful and inspirations for the Korea domestic science areas. So the PRP is the upgrade is NRP. The NRP is the going to the GRP. GRP is versus APRP. The APRP is to get the fund from the Asia Connect project. Yes, the global trend is, as you mentioned, is the data is increasing remarkably, and for especially the ASEAN areas has a lot of the smart brain, but we don't have enough computing resource to implement our thought and our research. This project is the main goal is to provide research platform distributed to make this the ASEAN areas is the upgrade about several the research areas. So this is the whole picture of this our project. A bandwidth distributed HPC, and the advanced countries is donated some servers and the experience of technology, and the beneficiary countries is the running or the implement their thought and their research and their code and something like that. So in case of we are two cases. So we are the three steps. Activity one is building a big data superhighway based on 10 or the NLANs for globally. And the second activity is building a distributed HPC platform based on the Kubernetes. The Kubernetes is a game changer to make the distributed environment, as you know. It's very powerful and very dominant technology. And the third thing is we deploy the pilot user case, AI and the bioinformatics. This is briefly the picture of the building of the big data superhighway based on 10. It is very important, but it's the harder things because the endpoint is the network configuration is not easy to is what we want. So we have this continuously the negotiation and the discussion with the local network operation center and the local network engineers. And we want to make this the high speed end to end the network environment. This is our the achievement through this 1 year our project. And then as we set up the TTN and make the fast guarantee and the set up this the persona, this is the network measurement system and how to the set up this manually. And this is activity two, is building a distributed HPC platform. We distributed KIST, NCI, and Circle IB University, Ferradana University. And finally, we want is a part of this GRP or PRP. So we are the building and the construction is the right side is the platform architecture inside. The left side is the what kind of the open source what we used. And at the bottom, the table is the hardware specifications. And this is our the activities is the last 1 year, and the workshop, MOU, and the set up the master node and the tutorial, and make the conference. And then this is the user management technology based on this blockchain technology. But it's just the prototype, the research. Currently, there is a user management is the ID federation technology. And then this is the very simple user the interface. We wanted to make this web portal, but it takes a long time. But just 1 year, and we make this very easy, the access point as the web the model. This is the activity 3 is the AI science and the bioscience. In case of the left side is the AI science case from Pakistan. The left side is the bioinformatics is the Malaysia case. They currently running their jobs on our research platform, and they made the efficient result. And this is in case of the running the distributed platform, the using 1 or 2 CPU, and then using 2 GPU machines. And the GPU machines is much better the result. I'm sorry. And this is the other achievement. What I mentioned before, there is some conference and make the video clip and the technical report and publication international journal and something like that. Just for 1 year. So our new challenge is the Korea domestic. In case of the Korea domestic status, the related 3 the project is the ongoing, the underlines. The first thing is the smart hospital development project. The title is the medical issues. The contribution point is a big data superhighway for inter-hospital for human and genome data centers. The second thing is Korea Rural Development Administration project. The title is Integrated Linkage System for the Agriculture, Big Data, and the Utilization Model. Our contribution point is to the running the crop models system or the research platform donated and building and support for the end user. They have the 2 types of end user. The 1 type end user is just the farmer. Just the farmer is the using the result of this crop model, and the user is agriculture researcher. They're learning their model on this platform. And the third thing is a Korea Environment Institute project. It's granted the last December, and the name is the establish an integrated impact assessment platform and the building of the ensemble of the multi-climate scenario based on the new climate regime. Very complicated, but the contribution point is building a container-based research platform and the self-storage. They want to do the several models is make the ensemble using the AI or something like that. So we can develop or the construction or set up the research platform based on the container technology. It means that it's a Kubernetes. And the store, the share, and the sending the big data, the using self-storage. And the last slide is the future plans. Is all of this contents is the result of our Asia Connect project, we want to continuously deploy this technology and expand the local point in Asia areas through the APRP working group. So our beginning step is just the collaboration is Australia, Malaysia, Pakistan, and we want to expand it to neighbor countries. And all of our using the managing and the operating, and we will do it through the Asia, the research platform, the working group. And the extension for third parties research areas is just, I assume there's three areas. The first one is smart agriculture. The agriculture problem is a big issue, is everywhere. So we want to run the crop model or the climate change model or the combining and ensemble model running as a smart agriculture on this platform. And the environmental research on the climate change as a pure and the climate change research area. The third thing is the cloud computing and the wireless communication areas is kind of based on the IT areas. The cloud technology is the core technology. And the wireless communication is, we know the 5G and the 6G environment is almost overcome this wired networks. So the wireless technology is very important to the long distance and the isolation areas. But so far, I tried to explain about the 1 year Asia Connect project, and it takes over the 5 minutes. I want to show the next presenter, and if you have any question or about this project and to me, and please is asking the break time. Okay, thank you for your listening. And I will introduce the next presenter is Ki-hyun Kim, and from KIST in Korea. He is one of the great partners to develop the core architecture of a research platform. Okay. Ki-hyun Kim, are you ready?
Yeah, I'm ready.
Yeah, so please go ahead with the presentation.
Uh, can you hear my voice?
Okay, it's where.
Okay. Uh, Hello everyone. My name is Kian Kim. I work at KIST. I'm charge of development platform in Asia Connect project. Today I will explain. Wait. Ah, okay. Today I will explain platform development in Asia Connect project. I will explain about the APIP infrastructure status. This figure is for the establishment of Science DMG in Asia Connect project. This project establishes Science DMG in Korea, Australia, Pakistan, and Malaysia, and connect distributed DTNs in Asia to establish Science DMG. After that, the APIP platform is developed using the DTM built in each country. Based on that infrastructure, we want to develop an AI research platform for use by researchers in various fields in Asia. This figure is this figure is a configuration diagram of equipment construction in 4 countries. As shown in the picture above, 4 servers and 1 switch were completed in Malaysia. And the picture shown above is a picture taken after actual construction. So we configure 3 worker node and 1 master node and 1 self-storage node. The GPU consists of 6 and the CPU is about 180 cores. The storage size is composed of about 55 terabyte. Uh, and Australia donated 1 DTN server, and Pakistan will donate 1 DTN server, and Korea donated 1 storage server, and SAP storage is built and connected with Malaysia master node. I will explain about how to develop APRP. Most of the API platform was built using open source. Open source platforms were installed for platform management, and Orchestrator was developed so that users can use them more easily. As open source used to manage the platform, Kubernetes, which creates and manages the most important containers of the platform. And the SAP storage are used for storage, and the Harbor is used as a private image registry. Uh, and System monitoring uses Prometheus and Grafana, and server logs and errors use Elasticsearch and Kibana. Open source is used by users to perform AI tasks or programming using R, MPI, PyTorch, TensorFlow, Python, and so on. Shown above, the interface basically provided by the platform is configured to utilize Jupyter Notebook. For all images, Jupyter Notebook becomes the base image. It also provides a command line interface. The reason why a Jupyter Notebook is used as a base image is that the environment that links the authorization system will be linked to Jupyter Notebook in the future. The structure of the AI research environment providing platform is as follows. The most important role among each component is by far the Kubernetes cluster module, and through this module, all functions are developed to operate on a container basis. From the user's point of view in this structure, it consists of login, computing, transfer, storage, and monitoring. The transfer part is transmitted using GTN, and the login is configured to login through the uh, kick lock. And I will explain in more detail about the computing and storage monitoring part. First, let's talk about the computing module. The computing module is describe a computing environment and a distributed computing environment. The individual computing environment is configured so that individual users are allocated the computing resources. They wanna create a container, and the created container image is created based on Jupyter Notebook. Jupyter Notebook is an open source web application that allows you to create and share documents, including live code, visualization, and so on. Uh, when user create a container, a web URL is automatically created, and if they enter through the URL, they enter the Jupyter Notebook UI as shown in the figure below. The AI coding interface that can be used inside provides various interfaces such as Python, TensorFlow, PyTorch, Keras, and MPI. Uh, Kubeflow is a free and open source machine learning platform designed to enable using machine learning pipelines to orchestrate complicated workflows running on Kubernetes. Kubeflow provides various frameworks for constructing researchers' machine learning models. The purpose of Kubeflow in this module is for distributed computing. Distributed computing environment provides massive computing using all GPUs in worker node. To use Kubeflow's distributed computing, you need a parent server and a worker server. Uh, and next, in order to perform the compute module, we need a system for sorting images. Hover is a tool for private image registries. In our system, it is used to provide images in private and public environments. In the case of private images, users store the image they create in Harbor, and the provided system provides an environment where users can share them according to the user's permission. The storage module consists of SAP and RU. SAP Storage is an open source software-defined storage platform that make it easy to manage storage servers with software. It provides object storage, block storage, and file system interface, and uh, the size of the current provided storage server is about 55 terabytes. And when configuring the storage, it is configured as object storage. Object storage enables data transfer with other users, and when object storage is configured, it is currently configured to link with other cloud platform to transfer the data I used to the cloud platform. Next is the monitoring module. The monitoring module used Prometheus and Grafana. So this is the graph configured in the currently configured monitoring system. I will explain about APRP currently status. This is the currently login screen of the API platform. All of the login related part is done, and the user will log into the platform. If you access the platform after logging
Hello,
in, you can see the
hello, 기현 씨.
following. Yeah,
예, 여기
hello.
네트워크 문제가 좀 생겨서 조금 기다려야 될 것 같아요. 잠시만
저
좀
근데
대기해 주시고 내 카톡으로 보낼게요.
저 근데 빨리 돌아가 봐야 되는데.
예, 잠시만 기다려 주세요. 지금 거의 몇 장 남았어요?
저 이제 거의 마지막입니다.
그러면 지금 약간 앞으로 가볼래요? 조금 더 하나 더, 하나 더, 뭐 하나 더, 하나 더, 하나 더. 그 다음, 그 다음 페이지, 다음 페이지, 다음 페이지, 다음 페이지. 요게 셉 스토리지구나. 요거 다음이 뭐예요? 요거
뭔
다음.
소리지?
앞에 셉 스토리지 토폴로지부터 해야 될 것 같은데, 여기도 시간이 만만치 않은데. 일단 요거 전에 페이지부터 하고, 예, 여기서부터 해야 될 것 같은데, 잠시만 기다려 주세요. 이거 호텔 네트워크에 한번 죽었거든. 그러니까 카톡으로 바로 알려줄게요.
네. 예,
기훈 씨, 지금
예,
시작해주면 될 것 같아요.
예, 예. 네. 어, uh, I will restart. Yeah, the storage module consists of SAP and Lu. SAP Storage is an open source software defined storage platform that make it easy to manage storage servers with software. It provides object storage, block storage, and the file system interface. And the size of the currently provided storage server is about 55 terabyte. Yeah, and we use the platform use object storage interface. Yeah, and the next monitoring system module. Yeah, this is the graph configured in the currently configured monitoring system. And then I will explain about the APRP currently status. This is the currently login screen of the API platform. All of the login related parts are done, and the users will log into the platform. If you access the platform after login, you can see the following UI. The menu is divided into four categories and consists of dashboard, container management, storage management, and node management. Yeah, and then the current state of APRP is not completely uh, complete. The UI part needs a version update to the previous version. Also, when calling APIs for storage and container create, get works normally, but post does not work normally. We are continuously working to resolve the issue, and it requires a little source code change, but it is solving the problem of being created beyond the limit of the GPU. When the APIP platform is completed, we plan to deliver guidelines on how to use it from the system administrator's side and from user side. And when we did the user test in the last tutorial, we saw that users needed a detailed guide on storage. Container storage has a separate folder for object storage, and it is thought that there is a need for guidelines to store data in that folder. Yeah. Thank you for listening to my presentation.
Okay, thank you. And Kian, thank you for your presentation. And we don't have much time to the communicated the Q&A, and we're
Okay,
gonna
okay.
start this next presenter. Thank you, Guillaume. And can
Thank
you hear
you.
me, Asif Plaza?
Yeah, can you hear me?
Yes, ready. And please go ahead. And he is the one of our the Asia Connect project partner is the from the Pakistan. He introduced the AI science and the related work. It's kind of the distributed HPC environment. Asif, go ahead.
Yeah, Moon, can you please share the screen? I cannot share the screen right now. Can you allow me?
Wait a minute. His material is not on the screen.
I'm not allowed to share my screen.
Uh, he cannot allow the permit to share their screen. Asif Plaza. Okay, to share your material.
Okay, can you see?
Okay, we can see.
Ah, okay, hello everyone. My name is Asif, and I'm the co-PI of this, the project, Asia Connect project. And I was working with the Moon and Andrew and Keon Kim and Ashila and all these guys on this project. Before that, I was part of the Sakraibia University as assistant professor, and currently I am working at Fermilab as a network R&D professional. And recently I joined here, at Fermilab in US. So I'm going to present about the AI science use cases that we deployed, by using the distributed HPC platform at Sakraibia University. So these are the contents that I'm going to give you the overview of the distributed HPC platform, which already given by the Moon, but I'm going very quickly. And then a roadmap of Sakraibia University for AI science, AI use cases, and the future use cases and expansion plan for the HPC. And finally, the Q&A session. So if we talk about the distributed HPC platform, this was already presented by the Moon, that this is the plan that we had before, that we are trying to build this distributed HPC platform among these 4 countries. So we have to build high-speed network connectivity among these countries. We have the computing resources, we have the storage resources, and by utilizing all those resources at the countries like the Pakistan and Malaysia, we are running some workloads, we are running some use cases over this platform. And these were the 3 activities that we planned, which already discussed by the Moon. I'm not going into the detail. But as far as my part, I was engaged in all those activities, but mainly I was engaged in, the presenting the pilot use cases by using this platform. So this was the, in the first activity, we built the path, and we connected all the sites, as you can see, by using the Tian network. And after connecting all the sites, then we installed the DTNs, which already mentioned. But the main thing here is I like to mention that we also separated all the DTNs, all the DTNs at the sites in countries by using the Science DMZ architecture. I think if you are familiar with our Science DMZ architecture, that it's separating the scientific networks and your campus networks, right? So we also followed some security policy. We did some TCP tuning on all those DTNs, and we also had configured the monitoring system by using the perf sonar. We tested all the sites, they were working perfectly. Now, this was all related to all first 2 activities. Now let's jump into our third activity, where we utilize the distributed HPC platform for AI use cases. So before going into that, I would like to discuss about the roadmap of Sakraibia University for AI science, means where I was working before. So if we talk about the AI research at Sakraibia University, we are basically promoting the research of AI by using our department of the computer science. We have number of courses that we are offering for students, like AI, machine learning, deep learning, reinforcement learning, and so on and so forth. The second thing that we have within the computer science department, we have a CREB center, which is called the Center of Excellence for Robotics, AI, and Blockchain. So that center is dedicatedly working on the AI research at Sakraibia University. And it has some facilities and infrastructure, but not too much. So therefore, we utilize this platform, distributed HPC platform. What is basically the purpose of this CREB center is to promote the AI research in various ways, like we are conducting the trainings of AI, machine learning, and deep learning. We are conducting some workshops, we are conducting some seminars, and our students are utilizing the resources to do some AI research. Hey, this is basically, you can see, this is our CREB center, and these are some workshops and seminars for students in AI and robotics. So these are we are also working on some distributed deep learning frameworks. Our researchers, our faculty members who are working in AI research, They are, also working on some distributed deep learning frameworks like the PyTorch, the distributed TensorFlow, and Horowitz. All these, the AI-related distributed workflows. So currently, if we talk about our resources, our challenges, and our future requirements at our Sakraibia University, obviously, it will be, we have the shortage of the resources. The biggest challenge, I can say that we have the shortage of resources to run, to execute, to test the large scale workflows at our, at our center, at our Krebs Center in Sakraibia University. So what we have the plan that we should have a dedicated, HPC environment. There we can, do the R&D. So currently, because of the limitation of resources, we cannot do, high performance computing and AI related research in our center. Therefore, we are going to utilize this platform, which is called the distributed HPC platform. And we are utilizing this platform for AI research and education purpose. So what research we are doing? We are doing some advanced AI and machine learning research. We implemented some algorithms over there, advanced machine learning and deep learning algorithms on this platform. We also, have opportunity to utilize this platform for teaching courses. I mean, suppose that we having some sort of lab works for students that can perform by using this platform. And we also can conduct some AI, machine learning, and deep learning workshop and seminars by using this platform. So this is how we can utilize this platform, and we already did. I'll show you that what use cases we perform on this distributed HPC platform. And this is the first use case where we trained the machine learning model using the SVM. We use the platform's GUI, and we use the Jupyter notebook, and we coded, and these are some results. I'm going very quickly because shortage of time. So we also not only trained the model, we also deployed the model. We use the fast APIs to develop and deploy the model that we already trained, and we verify that model. This is basically the model of some sort of diagnosing the disease, blood sugar. This is the model related to that. The other thing that we tested, and we in this platform, is the distributed deep learning. As I told, that our researchers are also working on the distributed deep learning. So we demonstrated also how the distributed TensorFlow framework can use on this platform. So these are some results that you can see. The third use case that we also worked with distributed MPI workloads also. Not only the TensorFlow, we also run the MPI related workloads. And the fourth is basically the as I told, that we are also teaching utilizing this platform for teaching purposes. So we created some labs where a student can perform the labs by using this platform. So this is how we utilize the platform, and these are some more projects that we are going to implement and we are going to test in near future. This is the aspects and an emotion recognition from the audio in Urdu language, and then this is the complete architecture of our model. And this is the Pakistan vehicle classification using deep learning approaches. Another project we are going to test by using this platform, and offline handwritten text, and convert the handwritten text into the to fill the forms automatically. And this is multi-object tracking for vehicles, right? So identify the vehicle speed, identify the vehicle numbers, which lane it is going. So all those things we are going to test by using this platform, the distributed HPC platform. So now this is how we tested the platform by using the AI use cases, by utilizing the AI, research from the Sakraibia University. Now, as far as the future use cases concerned, I have some suggestions for this platform, that how we can expand this platform, distributed HPC platform. Currently, we have the AI and the bioinformatics use cases. So how we can expand this platform? We can expand this platform for more scientific research and education purposes, right? how we can, implement some scientific workload test beds such as the CMS experiment. We have some test beds here at USA in Fermilab and other sites also. So we can research on, the CMS workloads and utilize the computing resources of the platform, distributed HPC platform. And then we can, also perform some large-scale data transfer of the CMS experiment by using the high-speed networks. And not only just utilize the high-speed network, but efficiently utilize the high-speed network by using the sense network orchestration solution. So we can, dig into more, that how we can expand this distributed HPC platform, not only within the Asia, outside of the Asia as well. So I have this plan. We can talk with the Moon as well in future. So this is the sense architecture which I'm talking about. Not only, we are using the high-speed network, but efficiently use the high-speed network by using this sense solution. Sense is basically the SDN-enabled network for science at exact scale. So we can automate, orchestrate the network, end-to-end network among, the sites, among the countries. Whenever we need to transfer the data, we have dedicated point-to-point, end-to-end, path from site A to site B to transfer the data to, do some CMS related experiment and work on the, data transfer related things. So this is the current experiment test bed we have in US using the Sense Rusio FTS interoperation system deployment, where UCSD, Caltech, University of Nebraska, Fermilab, and also we have some sort of the Sun part as well, through which we are, utilizing. By the way, that UCSD is deployed the PRP project over there, and we are using that, similar type of project that we are, we are using here in APRP. So same type of project we have at UCSD, and we are utilizing for CMS experiment, and we deploy the Sense, architecture over there. So this is how we can, expand our future, the distributed HPC platform, not only in Asia, we can expand it to the US as well. So if the Korea and Kisti wants, Moon wants, and other, having the opportunity of this thing. So that's all from my side. I hope I explain some sort of, the AI use cases on this platform. If you have any question, you are welcome. You can ask it anytime now.
Yeah, thank you, Asif Raja. Thank you. He comes from Chicago. He currently is working for the Fermi Lab in Chicago. So, Asif, we don't have enough time, then we have to skip this Q&A session, and every day question is through email. Thank you for your attending and presenting. And the next speaker is Asif Han from Perdana University. He's one of the project partner. He working for the bioinformatics areas. Asif Han, are you ready? Asif?
Yes. Can you hear me?
Okay, go ahead.
Okay, thank you. All right, let me.
Permit to share his presentation. Asif Han, already done? Okay. Please share your the material.
Okay.
Okay.
Fine. Okay. All right. Can you see the screen?
Okay, we can see. Please go ahead.
Okay.
I'm sorry for this. I can give you this for 10 minutes, please.
Okay.
Yes.
Well,
I'm
thank
sorry.
you. Okay. Well, thank you, Moon, for the invitation to present, and thank you to the audience, for joining our session. Now I'm gonna be fast. This is only 10 minutes. This is gonna be a little different. I'm gonna present or showcase to you from a user point of view, and and I'm gonna be a user from the life sciences. So someone from the life sciences to demonstrate to you how the platform that was explained by Asif Raza and built by Jung Hoon and other team members, how it's very crucial and important for the work that we do in biology. All right, so this might be a little different. So very quickly, an overview. We all know viruses are very important, and we are just coming out of COVID, and there's now a new variant. So I'm not sure if we are fully out of COVID, but we all know the importance of studying viruses. So this demonstration that I'm going to give you is for the study of viruses. Now, there are many lethal viruses. These are just some of the names of those lethal viruses, and we just went through SARS. So COVID was, caused by SARS. So we are very familiar with SARS. There's some others that also come and go. Now, because of the importance of these viruses, there's a lot of study that's going on. And one of the key areas of study is the sequence data, right? We want to sequence the genome of these viruses, and because that's the blueprint. And once we have these blueprints, we can study them, understand them, and come up with ways like drugs, vaccines, diagnostics to overcome these pathogens, these viruses, when there is an outbreak. All right, so this is just to showcase to you that the data is growing rapidly. In fact, this is not the latest plot. The latest plot, like for SARS, it was just a tremendous growth. In fact, what we collected in 2 years kind of, surpassed what we had collected over the last 42 years. All right, so the amount of data growth is just amazing. All right, and there are many data sources. So like this is not the data is not put in some place that's locked and nobody has access to it. The data is publicly available. It's free. You and I can, even use our phone and just access these data. All right. So this is just an example of a browser. NCBI is a very famous portal. You can go there. You can go to taxonomy browser. You can search for SARS. You can search for all viruses and you will get the data. So this is, all viruses few years ago. So this is not the latest picture, 6 million. Now we have like 50 plus million. And this is SARS, 55,000. And now I think we probably have 30, 40 million of SARS sequences, right? So there are data sources. And what's nice about these data sources is you can actually navigate through the lineage. You can go at a species level, you can go at a family level, at a genus level, or all viruses. So if you want to know all viruses, you click there, you get the whole data, right? So when we have, and there are a few other sources. I'm going to skip this in the interest of time. And so there's a lot of data. Now, our viruses compared to bacteria and all that, viruses actually mutate, change faster. So that becomes another reason why we want to study, pay more attention to these viruses, because they change and we can learn something about these mutations. And that's why this new variant of COVID that has been reported is as a result of these changes that happen more rapidly in viruses compared to bacteria. Now, there are three types of, changes. We have something called genetic diversity. This is the DNA, and the DNA can change, but not all changes at the DNA change at the protein level. Some changes do not change the amino acid or the protein. Some do change the amino acids. And then not all amino acid changes actually change the immune recognition. So you might have a change in amino acid, but it doesn't change the recognition by the immune system. Immune system can still recognize, but there are some that if they change, it will change the recognition by the immune system. So there is complexity. It's not simple, straightforward, get the data, look at the changes, and that's it. All right. So this complicates the situation. And it's these antigenic changes that result in immune escape. These new variants, they escape recognition from the immune system. We have something called original antigenic sin, where you get infected, you become immune, but when the next infection happens, actually it becomes worse. Like dengue 1 infects you, you are immune now after recovering. But when dengue 2 comes, actually it becomes worse. Because your immune system thinks it's dengue 1 and calls the memory cells the soldiers for dengue 1, but this is actually dengue 2, different. So it gets confused. So there's a little bit of a problem there, and it becomes worse because the immune system attacks the body. And then, of course, all this diversity, it's a challenge in vaccine design and drug design and diagnostic design, but more particularly for vaccine design. All right. And then, just want to show that why one change, let's say position 9, W changes to D something, all right? Why that change is kind of punctuated. Just one change can be a huge impact, because the immune system, it recognizes small parts of these sequences. So for example, it could recognize 1 to 9, 2 to 10, 3 to 11, 4 to 12, etc. We do not know which part it recognizes, but it recognizes parts. It doesn't recognize the whole, long sequence. So depending on which part, it could, let's say it recognizes all these parts, so you can potentially impact up to like, nine potential T cell epitopes. Epitopes are, or these are, epitope is the word to something recognized by the immune system. So it can impact up to 9 epitopes. All right. So we need to study them, study all these sequences, and there are two different approaches: alignment dependent and alignment independent. Alignment dependent, you take the sequences, you align them very nicely, and you look for those differences, you color code them. So it makes it very easy to find and study that. However, when you have a virus that's very, very diverse, sometimes alignment becomes a problem. But that's not the only thing. If you want to study all viruses, you cannot align them because they're all very different. You cannot take HIV and align them with flu because they are not the same. All right. So when you take all viruses, alignment, it becomes impossible. So when you have impossible alignment, how do you study all these changes? Then you need something called alignment independent approach. Now, here's an example of an alignment independent approach, and where we actually try to find something called minimal set, or study the diversity in a different way by reducing the diversity. So for example, we have 3 sequences that are very different because here you have LFF and here you have QLQ. So overall, these are 3 different sequences. And what we do is we break down, we create these what do you call these k-mers of each of these sequences. So we generate the k-mers, and then we compare the k-mers, and we notice all the k-mers here are the same, and all the k-mers here are the same. This means, and all the k-mers here are the same. This means actually the middle sequence is useless, it's redundant. We don't really need it because the information is already captured by the first sequence and the third sequence. So this is called minimal set. We can actually study the data, get rid of sequences that we don't even need to study, and reduce the data set to a minimal set. That's where we then look for vaccine design and all that. So that's an example of an alignment dependent way to study diversity. It's a bit different from the alignment dependent. This is independent alignment free, and so we use these approaches. And then we actually created an algorithm to take all the sequences, do this, and then come up with a minimal set, and then we can then study the minimal set. And then so we built a tool called unique min that is able to do this. And here we demonstrate that this is 4 million sequences. We remove duplicates because there are many duplicates. We get about 2 million sequences, so huge reduction. So the databases have a lot of duplicates also. And then from the duplicates, we were able to reduce it further, like 1.3 million, almost like a 1 million sequences were lost. This percentage is from the total data, so it's much less. But from here, you can see we managed to reduce it further. So this is an example of an alignment in a free method to reduce the data so that analysis can be done. Now the challenges, and here's the tool, but the key is that we struggled a lot with this. In order to make this tool happen, we actually had to spend like 7 months just for this whole work to be done on a standard computing platform that we had. We needed supercomputing platform, something like that Asif Raza and Jung Hoon has described, to be able to reduce this to actually 18 days. And recently we managed to optimize this further and reduce it to, if I'm not wrong, like 2 days. So that's a huge improvement. And this has happened thanks to the platform, the technology, this distributed platform that's available, now through this collaborative project. So that's it. Just to demonstrate how 7 months of work became 2 months, and 18 days, and from 18 days it became just 2 days, and the importance of computing. And now this enables us to ask more questions, deeper questions, and take the tool further and do all kinds of different interesting analysis. And the work has been published. And I just want to acknowledge the student, Lee Chun Chong, who was actually critical to move this project forward, and the funding agencies and other collaborators. Thank you very much. Over to you, Jung Hoon. I hope I didn't exceed the time.
Thank you, Asif Han. And I think his state is Egypt. So are you right?
No, Qatar.
Okay. Thank you. Thank you for the great presentation.
Thank you.
And we want to skip this Q&A session. And the next, the presenter is the Vincenzo Capone from Jiang in EU. And please let us see. I think it's already. switching rate within three.
Yeah.
The right side. Oh, yeah. The first film.
Okay. Thank you very much. So my name is Enzo Capone and I work for Giant. What I'd like to talk about is the LHC1 multi-domain service, which I think it's quite an interesting case of a large-scale global platform that can, infrastructure, sorry, that can underpin data distribution platform for high-performance data. Just a little bit of history. So where is this idea coming from? It came from the design of the data model for LHC experiments. The original model, this is quite an old picture, was a hierarchical model, so that you had CERN as the so-called tier 0, and you had the number of distribution sites around the world called tier 1, and then word for the physicists to do their analysis. This structure, so a very hierarchical structure. So pretty simple to a certain extent. And in fact, the first device by the international NRAN community was what is called the LHC optical private network. So basically, a dedicated set of links, star, with a star shape, where CERN was in the middle, and a few dedicated links around the world to connect the majority of these large sites. You can see here what was basically the very first version of this network service. Then reality happens. So basically what happened was that while the design for the LHC OPN was based on this little amount of data at the bottom, the vast majority of the data that was produced afterwards by the user analysis was overwhelmingly higher than the expected design. So this basically caused a change in the model, which made from a very hierarchical model to basically an anarchy model, because the predefined path for data distribution were not allowed to allow this very large scale of data anymore. So basically, the hierarchical model could get lost. Any other side. That's where this new idea came from. So a different kind of network infrastructure to allow for this large-scale data distribution. And this was the first logical model of the future LHC1 network. So basically having a distributed global large-scale infrastructure that could allow a flexible connectivity of sites with different kind of topology. This was the first step. So the first step was the idea of setting up a multi-point to multi-point LAN, basically. So based on layer 2 circuits. Well, it turns out that LAN and WAN are called differently for very good reasons. Basically, when you extend the distance of these links, local area network protocols and the technology don't really work very well. So the maintainability and reliability of this kind of solution was basically hampered by the very long distances. That's why the second version of the LHC1, which is the one we have now, was based on VRF, so virtual routing and forwarding technology. So basically, it was a distributed multi-domain layer 3 VPN. And this was the very first implementation. Then it grew over time. So this was another version from a few years later. And this is what it looks like now. So you don't look at the, don't even try to look at the detail. I just wanted to give you the the scale of how this service has now grown to basically cover mostly all over the world. It's present in almost every country, every continent except Antarctica, as far as we know. In Asia Pacific, this was actually the first implementation was from 2011, and even there now it's grown into a much larger infrastructure. Now, this you can see was one of the first data analysis that we did. This was only on the GEAN network, and I think it was in 2012. And you can see there were peaks around 12 gigabit. And this was last week, where peaks only on the GEAN backbone in excess of 600 gigabit. And we have seen during the data challenge a few months back, reaching peaks of 1 terabit on the GEAN backbone for only LHC1 traffic. Now, the experiment that was supported by this infrastructure were originally the one supported by the worldwide LHC computing grid. So the 4 big CERN experiment. But then over time, other experiments have been added, have been made eligible to use the infrastructure, like the Bell 2 experiment, and further on the Pierre Auger Observatory, the NOVA experiment, the Xenon 1 experiment, and very recently the Juno experiment. So the reason for this, for allowing more experiment was basically to make the life simple for especially big sites, because multiple scientific organizations. So now the problem is, if you have 2 sites that support multiple experiments and they move data between themselves, they would have been forced to basically split their traffic. So from the site A to B that are both on LHC1, if they want to move data for Atlas, let's say, that was allowed. But if they have to move data related to other experiments, in theory, they were not allowed to use LHC1. And that would have made their life very complicated in terms of their own routing. Was the reason why more experiments were added, because if you have to move data from A to B and both sides on LHC1, it doesn't really make sense to do this sort of fine-grained selection of what data can go and what data cannot. So this was a good idea, as I say, to make life easier for site administrators. These are now the NRENs and the region network and the providers around the world that support LHC1. As you can see, they are basically everywhere. I try to more or less give a resemblance of geographic distribution. As I've said, you can see from east, west, north, and South America, all around Europe, and the Asia Pacific. There is plenty of network that now support LHC1. The infrastructure doesn't only come with the configurations and actual infrastructure. It also comes with some very important assets, let's say, that kind of enrich the platform in itself. There is a wiki that explains all the ins and outs and the detail of the implementation and so on. There are network operators guides, how to, and best practices. There is a BGP filtering facility that allows a very fine control of the traffic for the ISP and also the sites. So basically it's based on BGP communities, and they can have a very fine-grained selection of sites they want to connect or they want to advertise. So it's very powerful. There is now a hosts all the as I said, there are also site connection guidelines, and very important, there is a personal monitoring infrastructure that is extremely pervasive. So basically, every site that is part of LHC1 has a personal node installed. So this makes for the troubleshooting a very, very powerful platform. One of the things that is, as I said, is important is the flexibility of this solution. You can see that we can go all the way from a site that has multiple routers, connected to an NREN that allows for the deployment of a dedicated VRF. We can go all the way to sites that have one single router connected to an NREN that doesn't provide that facility, but that can provide transit to the next NREN in line that can provide the VRF support. So basically, okay, so I mean, just to conclude, so I was saying the good thing is about the flexibility. So it allows a flexible access to the infrastructure, both to the sites and to the NREN. Of course, if you have a simpler configuration in terms of infrastructure, so like a single router, it requires a bit more complicated configuration on the side because you have to do, for example, source-based routing, but it's manageable. So to conclude, LHC1 provides a number of advantages. So as I've said, some of these are related to the very nature of it. Like, for example, it has a very short routing table if you compare to the global routing table, even of the RNA networks, which means you have you have an easier time doing troubleshooting because you have less site. It also creates a trusted environment, which is the main advantages of this infrastructure. Like, for example, it allows you to create or deploy a science DMZ without any additional effort, because if you have two routers, one connected to LHC1 and one connected to the general IP, the one connected to LHC1 can have a relaxed set of security enforcement. Like, for example, you don't need a firewall. You can do basic ACL-based security, which means that you protect your data flow, your large data flows from the firewall that usually is the one that kills long-distance, high-performance data transfer. On the other hand, you have a reliable infrastructure because, as I've said, you have a pervasive personal monitoring infrastructure. There are well-identified peering points, which means you have also routing stability, which is quite important because you minimize the problems related to routing asymmetries. And as I've said, you have a very fine control in terms of the connection and the advertisement that happen on LHC1 by the BGP community facility, which basically it means that you end up with an environment that gives you very high performance data transfer, but also a simplified troubleshooting, which basically give you also the opportunity of sorting out problems quite more efficiently and quickly than on the general IP. And that was it. Thank you.
Thank you. Thank you for your presentation. Okay. Thank you. We're going to discussing about the Q&A at the break time. And the Professor Nur Pisa, are you there? Professor Pisa?
Yes, I'm here. Sorry.
Yeah. Okay, are you ready? Are you ready to start?
Okay, so I'm going to share the screen.
Yes. Sorry for that. I can give you 10 minutes. Please, whenever you're ready, and go ahead.
Okay.
And give the permit is Nur Pisa, Mat Isa.
Okay, so I can, sorry, I want to find out this one. Okay.
Okay, we can see you.
All right.
Please go ahead. Yeah.
Okay, sorry.
And she is the professor of the related bioinformatics areas. His presentation is aviation infection diseases in the professor in the UPM from Malaysia. Please go ahead.
Okay. So I'm going to present on I'm going to share the presentation actually based on what I'm doing, and then what is still related to the HPC and also the big data. Okay, so basically my presentation title is Poultry Future Data Driven in the Fight Against Avian Infectious Diseases. So I'm covering the some of information on the poultry industry, the diseases that we concern, and then the prevention and control of the disease, and how the data-driven bioethics could transform poultry health monitoring in terms of the HPC application and some of the research highlight and development, which is I think more or less is related to Dr. Asif's presentation before that focusing on virus, but the difference is I'm focusing more on the poultry viruses. Okay, and then how from that information that we can have we can leverage the vaccine technology advancement way forward and how extreme the HPC is required. So basically, livestock is the second highest contributor to the value of gross output for the Malaysian agriculture sector, which is contribute about 18.7 billion, where the poultry itself is contribute around 16.8 billion. And Malaysia is self-sufficient for poultry meat and eggs, where Malaysia is consumption is among the higher in the world. Okay, so the rapid growing of poultry industry, however, not free from many issues and challenges such as high production costs and also emerging and re-emerging diseases. So to date, almost all non-major poultry diseases has been reported in Malaysia. So the most important thing is to prevent and control the poultry disease. Involve complete understanding of the interaction between the agent, host, and environment. And this can usually be achieved by the proper biosecurity, vaccination, and flock health management programs. So it's difficult and expensive to always maintain a high level of biosecurity. And those poultry vaccine and health management program are powerful tool in disease prevention and control. So this is the common disease that affect the poultry industry. You can see here that from the bacteria, fungus, virus, but our research here is more concerned on the virus infection. Okay, so basically, there are three important factors that involved in the prevention and control of the disease. The first, of course, the environmental factors, which include the proper biosecurity, good farm management practice. Okay, and then second is the host or chicken itself. Which is we need to implement the effective vaccination and monitoring program, and also again good farm management practice. And then the third one is the agent factors, which we need to diagnose the disease accurately, where we detect or we isolate the agent, and then we do the categorization. So in order to do that, so we need to do the effective routine virus surveillance and monitoring program, and also some farm also they do some effective treatment with the use of prudent use of the antibiotics or drugs. So basically, the whole process for accurate diagnosis is based on the conventional like history, clinical science, and also the detection and isolation of the virus or pathogen itself. And then, but later we move to the laboratory technique, which is more accurate, and then what we call more fast. Okay, so we focus more on the sequencing. Okay, so like Dr. As mentioned before, so from the sequence itself, we can tell many more information regarding to the health of the chicken itself also. Okay, so basically this is the ecosystem in our laboratory, where we have the lab that we carry the wet lab, wet work, which is includes the vaccine design and development, and also the disease diagnosis. So from that, we also do the sequencing here, and then from that data, so we move to the next lab. We focus on the dry analysis, which is here we can go for the genomic selection for poultry breeding, and then we can do for more information about the disease surveillance, and also the vaccine design and development program. And then we also have the facility for avian research and selection, where this one we reproduce the disease that happen in the outbreak, and then also we study about the vaccine potency and also the vaccine efficacy study. So how how actually the bioinformatics could transform poultry health monitoring, where we applied HPC here. So traditionally, bioinformatics is the science of store, retrieve, and analyze the large amount of biological information to solve a certain biological or biomedical problem. And this bioinformatics is normally include or involve different type of specialists or expert, not limited to biologists, but we include on the mathematics, computer science, right? So bioinformatics combine both computational and statistical approach in order to analyze biological data such as genome sequencing. So basically, this is the current model approach that what we call the broadened perspective of the bioinformatics, which is from the data is it can analyze not only for genomics, genomics, or only proteomics. Okay, so basically it can cross among these information, okay, to solve some of the problem. Okay, so basically so this is how actually we relate the HPC bioinformatics and also the traditional instrument research. So the ability of sequences in archive databases, for example, like the database in NCBI, as we mentioned before, and then some of the market price for sequencing of the sample has become affordable, and then the sequencing machine become more accessible. So means a lot of data has been produced. Okay, so this is where we need the HPC to collect all the data sample, all the information of the sequencing, and then from that we need to translate into more powerful information or powerful knowledge, right? And of course, in a shortest time. So, so this is some specific requirement to perform. Of course, we need the high quality of sample, and then we need the machine itself, and also the adequate computation structure and infrastructure and bioinformatic tools to analyze large volume of sequencing data are generated. So this is some of the work that we have done with bioinformatics. So we do the whole genome sequencing, sorry, for the viruses that isolate from the what we call broiler chicken. Okay, and then this sequence data also is what we call that a great promise to help monitor, identify, and track poultry flock health. So normally used to detect early infection. They may affect the productivity of the flock. So the data analysis allow for quick implementation of corrective management process, and the nucleic acid of the disease agent that are present in sample are decoded. Okay, so so basically from the data that get from the bioinformatics is extensively used in worldwide. For example, in China, they also use from some data from bioinformatic data actually to implement in their poultry health management system. Okay. And of course, for this sequencing information, so they actually is important in decoding the gene, so where we can know about the pathogen past, how long the pathogen persists in certain certain cases. Okay, infection, for example, and then can replace the contact vaccine and then investigate the mutation. And also all this information will point the way to vaccines, antibiotics, diagnostic kit monitoring program, and public health strategies. So beside the whole genome sequencing, we also do the RNA sequencing. So basically, as mentioned by him in 2017, the distributed computing environment allow functional annotation process of large transcript data to produce comprehensive information of the large transcript data. Right, so this is some of the example that we use RNA sequencing, and then data that we have from the estimated try when we infect different different chicken line with the infectious Wuhan disease or virus. Okay, and then from that we also do some of metagenomic profile, observing any evidence of the antimicrobial resistant gene released from the poultry farm to the community. So basically, this one is more related on the bacteria community in the farm. So and then beside that, some of our work also we do on the assess the impact on the efficiency of the poultry waste management, wastewater treatment. Okay, so this one is regarding more on the alpha diversity, microbial community profile. Okay, and then you also perform gene network of nodes and age of ARG to species, and also the species. Okay, so all those information we use to leverage the vaccine technology advancement. So we study the bioinformatics of pathogen genome, and then profile host pathogen interaction, pathogen genome editing, and also a combination of the reverse genetic technology. So we can see that what we call the technology in terms of the vaccine development are changed from the conventional to the next generation vaccine. So this is what we want to thank for the bioinformatics, because from that information that we managed to design the vaccine that's suitable to the pathogen that is impact to the poultry. So this is one of my what we call research or finding, where we use where we study the pathogen of the chicken. Here we use the full adenovirus, and then we study about the sequence, and then from that we able to design innovative vaccine using the CRISPR-Cas9 technology, where we shorten the time to what we call to get the sick and stable candidate vaccine. Instead, use the normal attenuation process, which need more longer time to produce the candidate vaccine. So way forward, it is important to detect any genetic diversity and changes in genetic materials of the viral and bacterial protein, as well as their host immune response to solve the poultry infection outbreak issue. So bioinformatics provide a great promise in monitoring, tracking, identifying, and protecting our country commercialized poultry industry, and the exponential growth of HPC technology will enhance the genomic surveillance protocol and enable researchers around the globe to combat against the avian infection diseases holistically and efficiently. So how extreme HPC is required? So basically it depends on the data and choice of the tool. And if you mention to the downstream analysis, of course, depends on type of the analysis. Is that de novo assembly? So we need the Linux-based server, and then for parallel amount of data and application, so we need to what we call, depends on the platform chosen, either 455 or MySeq. Okay, and then the objective of the study itself, either we want to focus on whole genome or transcript assembly, and then it would need multiple core CPU, better GPU, and huge RAM. So if that available reference genome, or we call the direct mapping, so maybe less RAM can work. Okay, and CPU 16 core for small classes. And then some study we need the GPU, which is focused on visualization and protein modeling. We include the graphic part. So utilize GPU power for processing and computation, for example, like molecular dynamic simulation. MDS has seen huge acceleration speed up due to GPU-based code. Okay, and then for example, Ember, Gromax. Okay, so all these major MD simulations have released their GPU-based parallel version for speedy simulation. So apart from this, many other algorithms have also been made parallel using GPU, like multiple sequence alignment, engine analysis software, population biology software. So with conventional application for fast graphic processing, and also with extended cavity parallel processor, GPU cards have been really helpful for bioinformatics. So of course, there are limitations and strength of HPC bioinformatics relationship. So the strength is, of course, the time to workload ratioability and cost effective, and also time saving in producing comprehensive amount of data. But the limitation, because we are life sciences experts, so we requires an IT background personnel to assist in managing the HPC environment, and also we require proper HPC facility, hence open for collaboration. and use as a case study for HPC performance, because we thinking about to collaborate with the poultry industry farmers or SME communities to actually to provide a solution in terms of the surveillance program, where I think we need the real-time data collection, and also the from that data that we can go for HPC for the large data analysis, okay, to open the opportunity to what we call to find out the biomarker to detect or to predict the disease in future in the poultry farm. Okay, so I think with that, thank you.
Thank you, the Professor Fija. And the next turn is the Susumu Date from the Osaka University in Japan. He gonna share the experience of DTN, and the session one, the last presenter. Yeah, it's yours.
I hope it works.
Yeah, it works well. You can take the 10 minutes.
Okay, okay.
That's really planned.
Hi, I'm Susumu Tate with Cyber Media Center. So today I'd like to talk about just update of the last presentation in APAN, Nepal. So this is so I'm from the Cyber Media Center, Osaka University. Basically, Cyber Media Center is a supercomputing center at Osaka University. So we now providing the two supercomputer systems, Octopus and Squid. Importantly, we are providing these computing services to the Japanese researcher, but international researchers like you can also use our supercomputing system using the framework of JHPCN. So JHPCN is actually the collaboration among the supercomputing center in Japan, and we are providing the CPU, GPU, and many different type of resources. And if we can set up the international collaboration with us, so you can use the supercomputing resources. So this is very important things. And last APAN, so I explained my plan about the DTN solutions on data infrastructure in Osaka University. So let me explain what is Onion and what is why DTN is required. So again, so actually in 2021, so we have a deployed supercomputing system named Squid. So Squid is basically the supercomputer for quest to the unsolved interdisciplinary data science. This is an acronym, the name of the supercomputing system. And through the provision of this supercomputer system, we are aiming to enhance the use of the AI, enhance the use of the supercomputing system by AI people. But now we are not worried about the use of AI supercomputer system by AI people, because AI people are already using the supercomputer system. So because actually we have providing the A100 GPU resources. Anyway, so so important point. So now supercomputing center is focusing on the provision of supercomputing resources to the HPC people. But the AI people always complain to me, okay? HPC people always think about the computing resources. Okay? But the AI people think about the data. So where data is located is very important. So that's why we designed the data infrastructure, so named Onion. So Onion is the Onion is a data aggregation infrastructure. So Onion is short for Osaka University Next Generation Infrastructure for Open Research and Open Innovation. So we use a small portion of our procurement budget of supercomputing system to deploy this data aggregation infrastructure. Thanks to the Onion, so now people, the researchers in Osaka University are enjoying the people, researchers in Osaka University can easily move the data from their department and their own department or research institution to our cyber media center supercomputing system. So before the supercomputer system, the AI people always complain. Okay, so data a supercomputer system just provides the data transfer system like SCOPy or other very command line, very simple way of data transfer. But now, so the campus user can move the data from the IoT devices to supercomputer system easily, and also the user can move the data from the scientific devices like telescope to the supercomputer system. That means actually we can increase the utilization of supercomputer system because the data is located in supercomputing system. That is our strategy. And the structure, oh, sorry. Actually, the Onion structure is very simple. Actually, the combination of the three different storage solutions. So one is a Nextcloud, one is a exascaler, so parallel file system, raster file system, and one is object storage. So parallel file system provides a high performance I/O. So read-write I/O performance is very good, and the object storage provides the secure management of the data. And Nextcloud provides an intuitive web interface. And through the synergy of these three storage solutions, now the supercomputer users enjoy the data movement on to the supercomputer system. But one of the problem was there, and actually now people can obtain the way of the moving the data. But the performance was always an issue. So now our users always say, oh, okay, performance. So Onion allows us to move the data easily, but the performance is the issue. So that's why actually in last APAN, so we just showed this plan. So now supercomputer center is located in Cyber Media Center, and so in the campus, there are many department and research institutions that produces a lot of data. And that's why, so we are now planning to connect the data, establish the 100 gigabit network to each research infrastructure, research institution and department. And on both sides of the Cyber Media Center and research institution, we are now trying to deploy the DTN, data transfer node, so that the researcher can move data very quickly. So this is the plan. And but this is just plan, but not yet. And so this from this slide, I'd like to just show the progress and update for the last APAN. Okay, so now we are designing the red onion from actual operation perspective. So red onion means research enhanced onion. Okay, so onion is just for the anyway, so red onion is a research enhanced onion. So this is still designing. So maybe I will change the design. So anyway, supercomputer center is here, the right side, and left side is research department or the research institution. And in each side, so DTN DTN servers are deployed, and in addition to the DTN server, we are now planning to deploy the SSD-based storage, so stage 4 staging and aggregation storage. So, and the actually basic principle of the design is simple is the best. That's why we just provide, we are planning to just provide the high speed data transfer services on the red onion the infrastructure. So if our design is completed, so I think the user can just choose the destination of the data transfer from the data sources. And so maybe the infrastructure, interface is very simple one. And if the data, in the case that a user transfer the data from the department A to the department B, maybe the medical school to the research department of the life science, and in the case actually all traffic going through the cyber media center, and on the way to the data transfer, so data is stored in our supercomputing center's object storage, and object storage can be connected to the parallel file system. So that's why the data is always located on our supercomputer system. So after that, our HPC user and AI user can enjoy the high performance of the computational resources provided by our supercomputer system. That is plan. So many scientific users just think that, well, high-speed network, so I mean the physical network, is enough. So because if we set up the 100 gigabits network, so science people believe that 100 gigabits throughput can be obtained, but that is not true. So that's why actually we have to check the performance of the DTN. So I mean whether the DTN can actually gain the performance. So that's why I'm now trying to test several DTN technology. Sorry, my time is over. So now actually this is a candidate, and now we are testing the local. We are now trying to investigate the performance profile of the several DTN from the actual perspective. So actually, we know that DTN as a service is a very good technology, but unfortunately, they don't have any good technical staff enough to operate the open source based software. So that's why we have to get the help from the Japanese company. So we are now thinking about which one is a very good in daily use of the DTN. And so this is so now still actually we are testing our red onion solutions in local area network, but we are now planning to have the same demonstration. The demonstration of supercomputing using the network between Japan and United States. This is actually the abstract of actually I submitted, and maybe if this is the slide of showing the supercomputing 19. So actually, at the time we used the 10G network, but in the case of supercomputing 2023, so we are planning to use 100 gigabit network from Japan to the United States. So I hopefully actually we are getting the same result, a good result in supercomputing 2023. So you see in 19, actually we gained the good performance of using in the case of using the 10G network use. Hopefully we get one very good result. Sorry, actually that time is running out and already over. So summary, this is the my upgrade. So now we are still designing the red onion, and for the purpose of the performance evaluation, so we are going to do the red onion demonstration in supercomputing. That's all. Thank you so much.